Okay, next up we have Arovella Therapeutics, ASX code ALA, with a market capitalization of approximately 173 million. Arovella Therapeutics is a biotechnology company focused on developing its unique inked cell therapy platform to treat cancer. Presenting today is Dr. Michael Baker, their CEO and MD. Michael, welcome back, and please take it away.
Thank you very much, Paul, and thank you very much to everybody for listening in today and for organising the event. We appreciate the time to be on here. So as Paul said, I am the CEO of Arovella Therapeutics. I'm very much looking forward to taking you through what we're doing in terms of using our iNKT cell platform for cancer treatment. For those of you that don't know us, we're listed on the Australian Stock Exchange under the ticker ALA. Next slide, please. I will be making forward-looking statements, so please do note the disclaimer. Next slide. Now, I'd like to start here with the strengths of the company. So as I've just noted, we're working on our iNKT cell therapy platform, which we're using to tackle initially blood cancers and coming up behind solid tumours. Our lead product is called ELA-101, which we're using to tackle what we call lymphoma or leukemia, and expecting that to commence fiscal year 2025. Now, the cell therapy sector has shown a lot of promise and revolutionised some forms of cancer treatment, but there are a number of challenges with the current form. And we do think that our iNKT cell platform goes a long way to solving some of those challenges. Now, over the course of the last 3 years or so, we've built an exceptional team, both management level and board level, focusing on drug development or cell therapies, spanning both Australia and North America. In terms of how we've built over the last few years, we've really taken a strategic approach by acquiring what we would consider the best intellectual property worldwide, and then developing around that. And we continue to take a lens on doing that, finding technologies that either enhance our platform or broaden its utility. So for those reasons, and the fact that we are one of a handful of companies globally working on iNKT cells, we see that we're well positioned for growth. Next slide, please. So just a little bit about our financials. As you just heard, we're capped at about just over 170 million. At the end of March this year, we had 15.3 million cash in the bank. So we're well funded and have money to reach key milestones. Great shareholder register with a lot of support at the top end from both institutions and high net worths. We've had strong share price appreciation over the last 2.5 years, despite the headwinds that have faced the biotech sector broadly. But of course, we're looking forward to welcoming new shareholders to the register that understand not just the risks that come with the biotechnology, but also the potential rewards. And the way we're positioned, the way we're setting ourselves up now, we're looking forward to continue to deliver shareholder growth. Next slide. So I won't go through every transaction listed here, but this is an overview of some of the transactions that have taken place over the last few years, particularly for early stage cell therapy technologies. And there are a range here: acquisitions, research collaborations, license agreements. But what I'd like you to understand is that the stage is quite early. So phase 1, phase 1B or preclinical, and one listed there at phase 2. And what we see often for these types of partnerships is there's an upfront payment in the order of $200 million or more, which is, again, demonstrative of how important large pharmaceutical companies value this particular sector. I will highlight one transaction there, second from the top. It was last year, December 23, AstraZeneca acquired another cell therapy company, Gracell, who are in phase 1B with data, and they acquired them for an upfront payment of $1 billion. So again, just highlighting the importance of the sector here for large pharma. Next slide, please. So as I've just mentioned, some of you may be familiar with CAR-T, some of you may not. In that top left picture, the way they currently do it is they actually take this type of therapy from the patient's blood. So it's actually the blood is taken, the immune cells are collected, genetically reprogrammed, and given back to that particular patient. What that means is every single dose is specific for that individual and must be manufactured. So what that means is we end up with very high costs, about $500,000 US per dose. It's complicated logistically. And because it's coming from the patient's own immune cells, we know that they're likely to be compromised because they have seen nasty things like the cancer and chemotherapy. A range of treatment centres are capable of this, which means many patients could be eligible to take this type of therapy but won't get it. And because it takes in the order of 3 to 6 weeks to make, patients can and do die while the therapy is actually being manufactured. And because it's being made every single time, we are actually able to see that manufacturing run failures do occur, increasing time and costs. If you can click forward, please. So Arovella's solution is we use a different immune cell. So it's not a T cell in this case. We use our iNKT cell. We take the cells from a healthy donor. We're able to genetically reprogram this immune cell, and we can give that to multiple patients. So the idea being that we're able to scale up manufacturing and that we're ready to dose patients in less than 1 week of being eligible to take the therapy. Next slide. So the way we actually are able to do this, as I said, it's by using a unique immune cell that others are not working on. So top right, we've got T cells. Bottom left, we've got NK cells. And this is just a snapshot of some of the immune cells in our body. Those two have been the most widely studied. Next, click forward, please. But as I said, it's what we care about most is the iNKT cell listed in the middle there. And there's a few reasons for that. I won't go into all of them in great detail, but we know we can give them from a healthy donor to many different patients. You cannot do that with a T cell without additional genetic engineering. We also know that they are the front line of the human immune system, so they're very potent and very strong killers, containing multiple anti-cancer killing mechanisms. And in terms of solid tumors down the bottom there, there's a range of properties they have, which we think will also set them up to be very useful in the context of a solid tumor. Next slide. So when we think about the commercial landscape, can you click through, please? We were very mindful that we didn't want to be in the T cell or the NK space on the left and the right there. There were hundreds of companies, thousands of clinical trials ongoing. So we carefully selected an immune cell where we could achieve the goal of having robust anti-cancer activity, but also a product that could be used off the shelf. And that's where we landed on the iNKT cell platform itself. And just noting there, all of the logos that you see in the middle circle there for the iNKT cells, that's everybody in the space working on cancer therapeutics. On the left and the right, it's not all of the companies. It is a snapshot. Next slide, please. So what we've been able to show over the last, or we've announced over the last couple of weeks, is that we've actually generated what we call a clinic-ready manufacturing process. And I would like to talk why this is so important. And just noting the grey bars at the top there. So the way we actually get a product into clinic, we acquire a technology, we generate preclinical confirmation. We then get what we call a lentivirus. So you can see that little bottom right circle with the bumps on it. That is a lentivirus. What that does is it actually, we use that to insert the DNA to make the CARs in the cell, and then we go on to manufacture. So I'm just going to talk about that from the left here. So that little blue person, we collect blood from that individual, would be a healthy donor. We isolate specifically their iNKT cells. And as I mentioned, we take that lentivirus, we use that to reprogram the cells, and that's where we end up with those little yellow and blue flags poking up, which are the CARs. If you don't know what they are or you're not familiar, I'd like to think about them as targeting missiles or homing missiles. Those cells then get grown up into the billions, and then they get stored, frozen, and ready for use. So in the bottom left there, we've got a range of the numbers that we've been able to hit using our manufacturing process. I won't go through them all, but we're pretty comfortable that it is now industry leading. And we know that the FDA is currently working through consultation for a platform designation, which we think we would be able to get. And the way we do that is using that lentivirus approach. If we would like to make another product to target a different cancer, we just get a different lentivirus to make a different homing missile, and then we can still leverage all this work that we've just completed. Next slide, please. It's just cut a little bit short, unfortunately. There we go. Next slide. I seem to have lost a few slides there, gentlemen.
Apologies for that, Michael.
No, that's okay. What I can do is I can quickly give some updates verbally on what we're trying to do. So major goals for the company now are to take our lead product, ALA-101, into clinical trials. So we're expecting to do that this fiscal year, I should say. So we're well on the way to do that. And there's a number of activities like performing the manufacturing run, writing up all the documentation that we submit to regulators like the FDA and the TGA. Once we get their approval, then we're off and running to start clinical trials. So we do have another program, and as I mentioned, using a different lentivirus, the grey bumped thing there. We actually have a product that we're using and developing to tackle both gastric cancer and potentially pancreatic cancer. So for that one, what we're doing currently is setting up the preclinical studies to generate the data to support that would be useful. And once we've got that preliminary data, we take that into animal models. And we've also got another armoring technology that we'll use to strengthen the entire platform. Now, I should point out, again, looking at those particular lentiviruses, as I said, because we can expand the utility of the platform to tackle multiple different cancer types, we are actively reviewing the landscape and evaluating a number of those that we could potentially bring into the company. So in summary, I think what we've got in front of us is a cutting-edge iNKT cell therapy platform to tackle a range of different cancer types. As I said, we are very comfortable that we're in a unique area of one of the most important fields of cancer treatment that we'll see moving forward. And we're very excited to take our lead product, ELA-101, into clinical trials in the not too distant future. And I'll wrap up there, Paul. That sounds great.
Thanks, Michael, and apologies again for losing those last couple of slides for you. Okay, got plenty of questions coming in. The first one, you recently announced the completion of the manufacturing scale-up. Can you explain how that is going? It's
an excellent question and a good slide to be stuck on. So this is that process here, actually. So to do this, there's a number of different steps that aren't necessarily depicted here and not quite an infinite range of parameters that you could test for each of those steps. So what we've successfully done is taken the process that was developed by the laboratory at Imperial College London, which is patent protected, I should point out, and we've managed to take that now into a commercial environment through our manufacturer at Cell Therapies and demonstrate that we've got a process that would support late-stage clinical trials. So what we've been able to achieve is phenomenal. And as I said, we don't have all the details of some of the companies because they're private, but we're pretty comfortable it would be very close, if not the leading manufacturing process to date. So very happy with how that's gone. And now we're looking forward to doing the manufacturing runs for the material that we'd look to take into clinical trials.
Okay, and a question here. Earlier information from the company had the ALA-101 phase 1 trial commencing second half 24. Can you update, please, FDA and IND and opening of the trial?
Yeah, absolutely. So what we've got to do now for that particular clinical trial is we manufacture the material that we use to take into the clinical trial. So that requires that it's a certain grade called GMP, which we're going to start very soon. And in parallel, what we're doing is getting clinical trial sites, key opinion leaders, physicians engaged, and we're seeing some good feedback for people that would like to be involved in the clinical trial. Once we've got the GMP material, we do some final studies that we call IND-enabling studies. All that manufacturing work and all those studies get written up into documentation that we take to the FDA or the TGA and ultimately then get approval for them to start phase 1 clinical trials. So we are aiming to get a lot of that work done in the next or before the end of calendar year this year. So we've certainly got a pretty aggressive timeline, but looking forward to getting a lot of that work ticked off. And we'll be announcing a lot of those things as they progress.
Another question here. Can you please discuss the difficulty or ease of manufacturing good quality products and also the question of maintaining the quality of store products without deterioration?
I missed the first question, Paul. It just broke up. Would you better repeat that?
Yeah. Can you please discuss the difficulty or ease of manufacturing good quality product and also the question of maintaining the quality of store products without deterioration?
Yeah, absolutely. I'll start with the second part there. So a cell therapy, it's a living product. So what we end up doing is we store it in liquid nitrogen, essentially. So it's incredibly cold, and we're very comfortable with what others have seen for cell therapy products in the past, that we won't have issues with degradation. Irrespective, you'll still go out and do stability work to potentially 3 to 5 years. So you can demonstrate that a shelf life of 5 years would be adequate. Now, in terms of a cell therapy, the manufacturing process is arguably the most important aspect of working on these types of therapies. That's why we've taken a very careful approach to setting this up after licensing the technology from Imperial College London. We've spent a lot of time on this. And as I said, I think the way we've been able to approach it and through what the team's been able to achieve for the specifications on the bottom left there, I think it's it's remarkable where we are positioned today. And what we also know about cell therapy is that the product is often referred to as the manufacturing process. So again, we're very happy that our process is patent protected. But what also comes out of this is there's so many different steps and so much internal know-how that we have generated that will also serve as a competitive advantage for Arovella, which is important moving forward.
And a question here, how many scientists currently work in lab, and are you hiring more?
No, it's a good question. So the way we work as a company, we are virtual. And for those of you that don't know what that means, we essentially have a group of individuals that essentially run particular studies or particular programs, and we outsource our manufacturing and our research and development through either contract research organizations. We still have a research agreement in place with the inventor of the technology. So our requirement for hands-on scientists or lab scientists at the moment is low. And as I said, I think the most important thing for the listeners today as well is by having the virtual model, we've been able to control our costs relentlessly. And so I think that's been pleasing to take that approach because we can turn things on and off as needed.
And just one to finish up, how well is your intellectual property protected from hackers and spies?
It's a good question. Look, I think we're doing all the things that we can do, and we have good cybersecurity policies that are being developed and in place already. And in terms of our patents, several of them are granted already. So from that perspective, we're covered in the best possible way. And of course, I think it's always just about training and making sure that we're protecting our cloud documentation systems and so forth as best we can.
Michael, there's a couple of other questions. Maybe you can answer those offline. That's all we have time for now. Thank you very much for your time today, and you have a great weekend.
My pleasure. Thanks, Paul. Appreciate the time. Thank you.